Hi guys! Today we are going to go over how to go through the process of protein synthesis. So what this means in the scope of what we've been talking about in class is we start with the DNA strand, we go through transcription to make our mRNA strand, and then we go through translation to make our amino acids. So today I'm going to run through a few examples. These are the same examples that you'll find on your worksheet under the example section. So fill these in as you go, and then use these as a guide to help you with the ones that you're going to practice on. So you're going to need two things to complete these problems. Obviously, you'll need your DNA strand from your original DNA, which I've provided here, and then you're also going to need a codon circle. So this is your codon circle. The one you are using is posted on Google Classroom. I would highly recommend that if you have the ability to print this out, you print it out. It's going to be a lot easier to reference as you're working on your screen. If you can't print it, then just split screen like I've shown you in class. So you have your assignment on one side and you have your codon circle on the other. You're going to need this to figure out your amino acids. So for right now, I'm going to put this off to the side. We will come back to this in a few seconds. But we'll start with our first example. So my example one, I have a DNA strand that was given to you. How do I know this is DNA? Well, there's some very important things, or some big clues, and the first one, biggest one here, is that there are T's. So anytime you see a T in your DNA, or your strand, you know it has to be DNA. So the first thing we are going to do is go through the process of transcription and make my mRNA strand. So this is transcription. And when we go through transcription, we know our rules from our notes. Like we talked about with replication, C always pairs with G. But remember, this is the weird one. When we have A now, instead of pairing like with T as we did in DNA, we're going to pair it with U. So A pairs with U. So if we go through my first strand and create my mRNA, T is going to pair with A. We still have A in mRNA. A now, if we were creating DNA, we would pair it with T. But because we're creating RNA, we pair A with U now. So now it's AU. And then we pair C with G. G pairs with C. G pairs with C. A pairs with U. And I'm just going to continue on. Alright, so I've completed my mRNA strand, and by doing so, I can now move on to the next step. So the next step here is going from mRNA to my amino acids. So what we're going to do, that process, going from mRNA to my amino acids, that's called translation. And if you remember, my amino acids, these are my building blocks for proteins. And as we discussed in class, proteins, I know we want to think about like meat and food, but proteins, remember, are not meat or food in this case when we're talking biology. Proteins are all the structural things that make you up. So we have pigment proteins that help to determine your eye, hair, and skin color. We have proteins that help to shape your bones and your muscles and all the things that make you you. So when we're talking about proteins, remember, we're not talking about steak. We're talking about structural things in your body. Okay? The other thing you probably noticed is you're like, wait a minute, we skipped tRNA. When I do these, I like to skip that row so I don't get confused, and I'll show you why in a minute. So I skip right to the amino acids, and then I go back and do my anticodons. So we're going to do that first. So when you do your amino acids, this is when you're going to need your codon chart or your codon circle. So I'm going to pull this baby over here, and we're going to look at my first codon. So if you remember from our notes from class, the codons are always in sets of 3. And I was super nice on your worksheet and already broke this into these nice chunks of 3 for you so you can see your codons really easily. So my first codon here, that first chunk of 3, is AUG. So I'm going to take my codon chart and I'm going to go A, because that's my first base pair. You start all the way in the center of your codon circle. So I start with A. My second letter is U. I'm going to move to the second circle. And my last letter is G. So I'm going to go to my last circle, which is the little letters, G. So the amino acid that follows those 3 letters is methionine. Now this is a mouthful of a word, so I like to, when I'm writing out my amino acids, abbreviate them with the first 3 letters. So the first 3 letters of this amino acid is MET. So I'm going to write MET, M-E-T. If you were paying attention during your notes today, what we talked about, AUG is also a special codon because it is my start codon. So every single sequence you will see in this class will start with your start codon. This is what signals our proteins to start being built. You need that start codon. Okay? My second codon, let's do the next one, was CCU. So I'm going to do the same thing here. This time my first letter is a C, so again I'm going to start in that inner circle. So I'm starting with C, and I'm going to just cover this up so we don't get confused. My second letter is also C, so I'm going to move out to the second circle, C, and my last letter is a U, CCU. So this gives me proline. So again, I'm not going to write out the whole word. If you would like to, you can, but I generally just write out the first 3 letters. So proline, P-R-O. Okay. We can continue on. I'm going to replace this codon chart so we don't get confused with what I've circled with a blank one, just to make our life a little easier. And we'll do the next few. The next one, ACU. So I start with A on my innermost circle, follow it to C, lastly get to U. So ACU gives me threonine. So I'm going to abbreviate THR. My next codon, CCC, again, CCC. This gives me proline again. Wait a minute, you're saying. We already used proline here, and my sequence was different. Yes, that is true. But you will notice that we have a lot of repeats. So in the case of proline, CCU, CCC, CCA, and CCG would all code for the amino acid proline. So it's okay if you have different codons that get you to the same amino acid. Next codon, AGA. A is my first letter. Start in the big letter in the middle. G, second letter. Let's go to that second row of letters. A, that brings me to arginine. Again, I'm just going to write the first 3 letters. ARG. And finally, my last one, UGA. U is my first letter, so I'm going to use the big inner circle. G is my second letter. A is my last letter. That, again, brings me to a special codon. It is my stop codon. That tells us that the protein has ended. So again, just like we always start with the start codon, we are always going to end with my stop codon. So now we have done the process of translation. At this point, we can go back and fill in our tRNA or our anticodon strand. And I'll show you why this gets confusing and why I do it in the order I do in a second. If you remember from our notes, the tRNA or anticodons are the ones that bring the amino acids, they're like the forklift, that bring the amino acids to the ribosome to help string them together. So the anticodons have to have the opposite of what the codons have. So we're going to base pair again. We are still creating RNA. So RNA is still going to use our base pair rules of C with G and A with U. So I have an A up here. I want to pair with A. It's going to be paired with a U. U is here. U pairs with A. And G pairs with C. If I continue using those same base pair rules, C pairs with G, and so on. And I'm just going to continue on. Okay, a couple things you should be noticing. If I compare my tRNA strand back to my DNA strand, you'll notice it's pretty much the same sequence with one exception. Anywhere we had T's up here in DNA, we have replaced with U's. Everything else should be the same. The reason why I do my amino acids before I fill in my anticodons is because you need to make sure that you are looking at the mRNA to create your protein. If you look at your tRNA strand to create your protein, you are going to get the wrong amino acids. So let's take a look at why. My first amino acid needs to be my start codon. If I use AUG, that brings me to that start codon, which is the methanamine, the crazy one. But if I use UAC and do my codon chart that way, UAC, that brings me to tyrosine. That is not your start codon, so that is incorrect. Using the tRNA strand is going to mean that you get all of the wrong amino acids. So I like to do my translation after I complete my mRNA strand so I don't get confused, I don't use the wrong letters. A big warning sign for you that you're using the wrong strand. If you start your amino acid sequence and it doesn't start with this MET, you're using the wrong strand. Every single amino acid sequence you create in this class should start with your start codon. Okay? Alrighty, so let's do example 2. I'm going to do this one a little bit more quickly just so you can see another example. We're going to start by creating our mRNA strand based off of my DNA strand. So DNA, I have T, it's going to pair with A pairs with U, C pairs with G, and remember we're creating mRNA this time. RNA uses U instead of T. And I'm just going to continue on. Okay, So I did transcription. So again, going from DNA to RNA is transcription. Now, again, I like to go from mRNA right to my amino acids so I don't write the wrong thing down. So I'm going to do that again. mRNA to your amino acids, which link together to create your proteins. This is translation. So again, I'm going to use my codon chart. AUG is my first codon. A, U, G. Hey, good news, guys. I'm starting with my start codon, the MET. So I know I'm using the right strand. My next codon, AGG. A, G, G. First codon letter, second codon letter, third codon letter brings me to arginine, ARG. My next codon, UCU. U is my first letter of that codon, C is the second letter, U is the third. My codon, or my amino acid is serine. GCC, GCC, alanine, so ALA, AUC, A is my first letter, U is my second, C brings me to isoleucine, so ISO, and lastly, UAG. Looking at my codon chart, UAG. Yay, it's my last codon, and it was a stop codon. Again, other ways to check. If you're not starting with a start and ending with a stop, you are probably looking at your wrong line. So now I'm going to go back and fill out my anticodons, or my tRNA strand. So, I base pair, again I'm creating RNA, so I'm using that A pairs with T, G pairs with C. I'm sorry, A pairs with U, G pairs with C. Look at me, messing up too. So A pairs with U pairs with A, G pairs with C, and I can continue on that way. And I am done. Now, can you do the tRNA before you do the amino acids? Absolutely. There's no reason that you have to skip it. For me, I just mess up less. It helps me out a lot if I go right to my amino acids instead of doing the tRNA first. I know that I've gotten things wrong because I've looked at that wrong strand. So if I were you, just to be on the safe side, skip to your amino acids first and then go back and do your mRNA strand. So now use the examples that I just went over to help you complete your 4 practice problems at the end of your worksheets.